there are a couple of things that you probably noticed on the study guide that were officially kind of part of the last unit that we didn't quite cover that we should cover to just kind of tie up some loose ends here. Just about the real nuts and bolts of neurons and how neurons communicate with each other before we get into like the big picture of the nervous system. So we are gonna go back and just tie up a few loose ends about the synapse, how one neuron communicates with the next neuron across this chemical synapse where you have, let's say this is our presynaptic neuron here. Here's our postsynaptic neuron. If you remember, there were receptors on the postsynaptic neuron. We had our presynaptic neuron had vesicles with neurotransmitter. And when the action potential reaches the end of this presynaptic neuron axon, what happens?
Sodium channels open.
Calcium channels open?
Calcium, yes.
Calcium channels open, calcium comes in. Then what happens next?
Exocytosis.
The exocytosis of the neurotransmitter, which is then going to go and bind the receptors there and cause some excitatory postsynaptic potential or some inhibitory postsynaptic potential. But what we didn't really talk about is, if we just left this situation like this, this neurotransmitter is just going to keep exciting the receptors, right? Even if this action potential happened, we had this exocytosis, we send the message to here, but we don't want the message to just stay turned on, right? Again, that's kind of like breaking off the key in the lock. It's like you send this message in, but we need to clear the synapse of the neurotransmitter so the message is stopped and waiting for the next round. So we're going to talk about our clearing the synapse. How is it after one of these synaptic events where we release neurotransmitter into the cleft? There's a message that's received on the postsynaptic dendrite or soma. How is it that this neurotransmitter is cleared out so the message stops and we're ready for the next signal? What are we?
Phosphodiesterase.
So that happens inside the cell. So it's the same basic idea. It's, the phosphodiesterase was about stopping the signal and second messenger transduction. But that's happening inside the cell as part of the second messenger pathways. Here we're outside the cell and we're clearing the neurotransmitter that's in the cleft between the cells.
Doesn't it just diffuse out, or is that wrong?
So that's one possibility, is just diffuses out. One possibility is it just diffuses out. So that's one possibility. And we're going to talk about, there are obviously going to have to be other cells that are maintaining the extracellular environment. So you don't just have random neurotransmitters floating around and causing mischief. So we're going to have these glial cells that are kind of vacuuming things up and cleaning things up and making sure that the extracellular environment isn't getting full of stuff that it shouldn't, and that all the levels of everything are balanced properly. So that's one possibility. Another possibility, though, is very similar to what Jambo was talking about. In phosphodiesterase, how did phosphodiesterase stop the second messenger signal?
It would eat up the camp.
Yeah, it was like an enzymatic breakdown of the messenger. So that's another possibility here is we can have enzymatic breakdown of the neurotransmitter. A classic is acetylcholinesterase. And a lot of acetylcholine synapses, there's actually an enzyme that's actually, it's anchored on the membrane that chews up the acetylcholine. So after the acetylcholine gets released and starts doing its job, it's now getting broken down by enzymes in there, so it starts to disappear right away. One of the drugs that is prescribed for Alzheimer's is donazepam, is actually an acetylcholinesterase inhibitor. Acetylcholine is involved in a lot of circuits in the brain that are involved with like attention. We'll talk about nicotine. Nicotine is an acetylcholine agonist. It's part of why you kind of feel more focused if you're smoking a cigarette or something. When people have Alzheimer's, they often give them this drug that blocks the enzymatic breakdown of acetylcholine, allows more acetylcholine in the synapses, and seems to help with kind of focus as people are getting into the dementia and stuff like that. So enzymatic breakdown is another possibility, but it's kind of wasteful. You're making the stuff and then you're breaking it down. What's one other way to, Wendy?
It gets back into the the pre-synaptic
Yeah, so reuptake.
reuptake, yeah.
Reuptake into the presynaptic terminal. So a lot of the synapses actually have little pumps going that are actually recycling the neurotransmitter so it can be reused, which again makes sense. It's like you release it, but then let's vacuum it back in and repackage it and have it ready to go again so we don't have to like make it all from scratch again. You know, it's like the classic, reduce, reuse, recycle. So that's part of what's going on here.
I have a quick question. Which one's like more likely to happen? Because like with reuptake, that would require energy, right? For like
they all happen in different cases. These are all ways of clearing the synapse.
Mm-hmm. I was wondering which one is anyone more likely to happen versus the other? Or?
No, I think it depends which particular system you're looking at. Okay. For acetylcholine, it's acetylcholinesterase, which is breaking it down in the synapse. There is usually like kind of vacuum back up. In fact, when you think about, what are SSRIs? SSRIs are like a really super common drug people are prescribed. What is an
a serotonin like reuptake inhibitor or something?
Exactly. This is a selective serotonin reuptake inhibitor. This would include like Prozac, for instance, fluoxetine. A lot of the more modern antidepressants act by blocking the reuptake of serotonin back into here. So in that case, you have your release of serotonin into the synapse, and normally it would just get kind of vacuumed back up after it's been released. But if you're on an SSRI like Prozac or something, then you block this reuptake, so you end up having an increased amount of the neurotransmitter that ends up in there. And exactly why that makes you less depressed is not clear. we know that that is the kind of direct effect of the drug, is blocking the reuptake. But then you have an increase of serotonin. It seems to downregulate the amount of receptors to serotonin. We've talked about if you have like increased like levels all the time, you tend to kind of lower the number of receptors to kind of make things less sensitive. But then for some, there's some downstream rewiring, and like 3 weeks later, people often feel less depressed. So we know empirically that they work as antidepressants, but you know, a lot of times people start getting into this super oversimplification about serotonin being, you know, this mood drug or something. But it's the way these antidepressants work is way more subtle. It's like, again, it takes about 3 weeks after you start this process before the drugs really start having their desired effect. For people where you're trying to get a really fast like if someone is like acutely suicidal, a lot of times there's a problem. I mean, people can off themselves before the antidepressant effects kick in. What are ways to try to have more of an instant antidepressant effect?
Socialize, exercise more, do all the normal stuff.
Alcohol.
No, so one of the ways it's still used is like ECT, electroconvulsive therapy, electroshock. Basically putting a current across your brain. And that actually is effective for kind of acute depression, especially someone who's like acutely suicidal. It can be useful, so they feel better like right away. It's not as barbaric as it used to be. They're much more skillful about how they apply it and how, and about putting things in so you're not, but it's still used. It's not like some, there's some things from the past that are ridiculous, but this one's still used. One of the more recent things is ketamine. Ketamine, which is a dissociative anesthetic, is also a, people use it out, lower doses to go out dancing even. But it seems to have kind of a more immediate antidepressant effect. But the classic SSRIs and antidepressants tend to take a while. They take a few weeks before they tend to kick in. So this is stuff you should know. Clearing the synapse.
I just have one quick question about that. So you said all of these can happen, just depends on where it is in the system. Does that mean these three things can happen at the same time as well?
Yeah, for sure. For sure.
Okay, thank you.
Okay. Let's see. So that was one thing I wanted to cover, was clearing the synapse. The other thing I wanted to do was just talk a bit about some of the most common neurotransmitters and that we're going to be seeing, just so you have a sense of them and you kind of have a bigger picture. There are a lot of different neurotransmitters. I think there's over 50 at this point that people have named or discovered. There's a few important ones that you should know about specifically. So let's talk about that a bit now. And we're going to talk about some of the main neurotransmitters. We'll also talk about some of the main classes of receptors for some of the main neurotransmitters as well, because that's going to actually be important as we get further into our discussion of the nervous system right now. So one neurotransmitter that I've mentioned before, acetylcholine, usually abbreviated as ACH. It's kind of its own class. Acetylcholine is going to be, it's a very common neurotransmitter. It's going to be important in the central nervous system, you know, particularly like it's been implicated in attention and things like that. It's also an important motor neurotransmitter. It's the thing that's released at all your skeletal muscles to excite skeletal muscles. It's excited, it's released at smooth muscle, particularly as part of your parasympathetic system. So it's a really common neurotransmitter. And there are two main classes of receptors that you need to know that bind acetylcholine. I should mention the adjective for things that deal with acetylcholine is cholinergic. So when we talk about cholinergic receptors or cholinergic pathways or cholinergic this or that, just means related to acetylcholine. So we're going to talk about now our two different classes of cholinergic receptors. And this might be easier just to type out so I can do it faster and it's more legible even. So there are going to be nicotinic cholinergic receptors. There's going to be muscarinic cholinergic receptors. So these are our two main kinds of receptors. They get their name, this nicotinic versus muscarinic. Those names come from endogenous ligands that will bind specifically to one or the other. Nicotinic cholinergic receptors, you know, you can probably guess from their name, will bind nicotine. Muscarinic cholinergic receptors bind muscarine, which is actually, it's found in kind of, you know, if your classic, if you've hiked around much at all and, you know, on Mount Tam at the right time of year, you've probably seen Amanita muscaria, the mushroom with the big red cap with the little white stars on it. This binds these muscarinic cholinergic receptors. Nicotinic cholinergic receptors, these are directly gated. These open up cation channels. These basically let sodium in. So we talked about directly gated versus indirectly gated channels before. So if you see some synapse and it has nicotinic cholinergic receptors, you know that it's an excitatory synapse. You're going to release acetylcholine into the synapse, and you know you're going to have an excitatory postsynaptic potential, because when you bind a nicotinic cholinergic receptor with acetylcholine, it's going to let sodium in, and the membrane potential is going to go up. The muscarinic ones, on the other hand, are indirectly gated. You know, second messengers. We call metabotropic receptors. What does that mean in terms of the response? If you have acetylcholine bind at a muscarinic cholinergic receptor? What's the response going to be?
Don't we not know?
Exactly. We don't know. It really depends on whatever that second messenger pathway is. So that's going to be important. Like when we look at skeletal muscle cells that have nicotinic cholinergic receptor, we know that those neuromuscular junctions are always excitatory. Whereas when we look at the parasympathetic motor, where we also have neuromuscular junctions with acetylcholine, but with muscarinic receptors, it really depends on which one you're looking at. It might excite the target or it might inhibit the target. We don't know. So it's important to pay attention to whether they are muscarinic or cholinergic receptors, because that's going to tell you a lot about the behavior of that synapse, whether it's excitatory or whether it has more of a broad possibility for what is going to happen there.
Can it never be inhibitory, or is it always like
Nicotinic?
the muscarinic?
That can totally be inhibitory.
Okay, that's what
Yeah, no, that's the whole point, is because it's a second messenger pathway, there are many different possibilities of what's going to happen. And you might end up just turning on enzymes or something. You might not even open a channel at all. Who knows? All right, so acetylcholine is an important one. The next group, we're going to talk about a group called the biogenic amines. The biogenic amines, these are derived from amino acids. So this is, on your exam, I actually showed you a couple of these. One version of these, so this is the big picture. One type are the catecholamines. These are derived from tyrosine. So tyrosine is just an amino acid, and then a couple of enzymatic steps away from tyrosine, you could end up with dopamine, which a lot of you have heard of. The two main ones that we are going to be looking at, we'll see dopamine a bit when we get into the midbrain and looking at Parkinson's disease and stuff. That's all about dopaminergic neurons. Dopamine is also very much tied into a lot of these kind of reward pathways, often tied into addiction and stuff as well. Epinephrine and norepinephrine. This is often just called epi. This is usually done NE. If we were in England right now, we would call them by different names. What would we call these if we were in some other place?
Adrenaline?
Adrenaline.
Exactly. Here we have the catecholamines, and the catecholamines would be adrenaline and noradrenaline. These are just synonyms. So epinephrine is what we call it, but you can also call it adrenaline. Adrenaline and epinephrine are synonyms. Noradrenaline and norepinephrine are synonyms. In general, they tend to bind to the same receptors. They are both going to be important players in the sympathetic nervous system. The adjective for things that are referring to epinephrine or norepinephrine, the adjective is adrenergic. This is why it's important to know that they're also called adrenaline, right? Because the adjective for something that binds epinephrine is adrenergic, which wouldn't make any sense unless you realize that epinephrine is also called adrenaline. So adrenergic means relating to epinephrine or norepinephrine. There are a large variety of adrenergic receptors. There are alpha adrenergic receptors. And there's a couple. There's alpha 1, there's alpha 2, there's beta adrenergic receptors, and there's a couple of those. When you have drugs that block these, like for instance, one of the common drugs that's prescribed for high blood pressure is a beta adrenergic antagonist. So these beta adrenergic receptors on your heart muscle will speed up your heart and increase contractile strength. So if somebody has high blood pressure, you're going to want to block that. You want the heart to stay slower and kind of help keep the blood pressure down. So you can have a drug that is a beta adrenergic antagonist. It's blocking the beta adrenergic receptors. So these adjectives are used in describing the different kinds of pharmaceuticals, right? If I have a drug that I'm going to put in your eye to block pupil constriction so I can shine a light in your eye in order to do an eye exam, I need to block these cholinergic receptors so the pupil doesn't constrict, and then I can't see what's going on inside. So I give little drops that are cholinergic antagonists, right? A cholinergic antagonist would block activity at the cholinergic receptors. If I have a drug that I want to stimulate, like if somebody has an asthma attack, part of the sympathetic response is to dilate your bronchial passages. So you can give somebody an adrenergic agonist if you want to stimulate the sympathetic nervous system, as we're going to talk about. And that would be actually binding and stimulating the adrenergic receptors so your bronchial passage is open so you can breathe again in the moment. So these adjectives, adrenergic, cholinergic, are used to describe the receptors. They're also used to describe the pharmaceutical, the drugs that will interact with these receptors or are mimicking these different kinds of neurotransmitters. We'll talk, for our class, you don't need to know exactly the details, the localization of alpha versus beta, alpha 1 versus alpha 2 and everything. As you get further on in your kind of clinical training and pharmacology, you'll learn much more about the specifics. Some of these are more localized on certain tissues in your bodies than others. Some of them bind both epinephrine and norepinephrine, or more primarily one or the other. One of them even is kind of counterintuitive. One of them is a post-synaptic receptor. Let's talk about that just briefly because it's important just to, I think about. Why might it make sense to actually have, not a presynaptic receptor? Why have a presynaptic receptor to the same neurotransmitter? So, normally you have the receptors on the postsynaptic membrane. The neurotransmitter goes and sends a message. Why have a receptor on the presynaptic membrane?
To initiate reuptake or something? Would it?
Or to inhibit, to stop the release?
Yeah, exactly. So it's kind of like a feedback message saying we got enough neurotransmitter in the synapse, we can stop releasing it. So you can actually have these presynaptic receptors to the neurotransmitter that help regulate the amount of neurotransmitter that's being released into the synapse. What that means is sometimes you get these kind of counterintuitive responses. There are adrenergic agonists that will actually will decrease the overall effect of kind of adrenergic synapses. Normally, if you think that I'm releasing an agonist to a receptor, I'm going to have more activity at that receptor. But instead, if you're releasing these special pharmaceuticals that actually are targeting, I forget, I think it's alpha 2, I'm forgetting now exactly which one it is. The particular, if you have an agonist to this presynaptic adrenergic receptor, you actually are decreasing the amount of activity at these adrenergic synapses because you're telling this thing, oh, there's already enough there, you don't need to release more. So just kind of putting out there, the reality gets more complicated than the initial story we were talking about. Does that make sense?
Can you repeat it one more time, please?
Let me maybe do it prettier. So this is like, let's say, so these are both gonna bind the neurotransmitter. And normally this releases it and it will bind here and send a message to the postsynaptic neuron. But also it's binding this one, and that is telling the presynaptic neuron, there's already enough neurotransmitter in the synapse, let's stop releasing, we've got enough already. But if you give somebody some drug that preferentially just binds here, that actually is telling this, oh, there's no reason to release more neurotransmitter. And so this drug that we just put in there is actually, even though it's an agonist to the adrenergic receptor, it actually is inhibiting activity at the synapse by turning off release of the neurotransmitter. So it's kind of counterintuitive that an agonist is actually inhibiting the activity at the synapse. But because you are targeting the specific presynaptic receptor, that is indeed what happens. So, does that make more sense?
Yes, thank you.
Okay. Okay. So we're talking about the biogenic amines. Definitely, we are going to be seeing a lot of epinephrine and norepinephrine. Again, norepinephrine is typically the one that's used as a neurotransmitter that's being released into synapses. Epinephrine, on the other hand, acts more as a hormone. It's going to be released by the adrenal gland, enter into the bloodstream, and just bind receptors wherever they are. This is probably as good a time as any to set up the difference between a hormone versus a neurotransmitter. They are both signaling molecules that bind to receptors on cells and cause some effect at the target cell. How are they different from each other?
When hormones go into the bloodstream, whereas neurotransmitters don't.
Exactly. So for instance, I might have, let's say here's synapse 1, and here's synapse 2, and here's synapse 3. If I'm talking about neurotransmitter, these might all be serotonin synapses. Or epinephrine, actually norepinephrine, or whatever. If I release norepinephrine here, it's going to bind receptors here, but neither of these are going to see it. If this one's releasing neurotransmitter, it's just affecting this particular synapse, and neither of these see it. So neurotransmitters are really localized to the specific synapse where they're being released. And only the receptors on the neuron that's part of that synapse are going to get the message. Hormones, on the other hand, these are typically released into the bloodstream. And basically, all cells with appropriate receptor will respond. So this is just a blanket message that goes out to everywhere. So like example with adrenaline, think about if I am releasing noradrenaline, I can like use my sympathetic nervous system to dilate my eyes, and just dilate my eyes, or speed up my heart, and just speed up my heart, or dilate my bronchial passages, but just dilate my bronchial passages. It can be a specific thing, but the other circuits that use that same neurotransmitter are not responding. If I dump adrenaline into my bloodstream as a hormone, now every cell that has that receptor is going to respond. I'm going to have my heart speed up and my eyes dilate and my bronchial passages dilate and everything getting ready to deal with some, the adrenaline rush, dealing with the fight or flight reflexes. So everything that has the receptor is all going to respond. So it's, whereas a neurotransmitter is just something that's happening in a specific pathway from one neuron to the next neuron. Does that make sense? They're both signaling molecules. They both bind receptors on the target. Hormones are this general broadcast out into the entire body, and every cell with the receptor that is the right receptor will have a response. So that makes more sense? All right. So, so epinephrine is usually the hormone. Epinephrine or adrenaline is going to be released out into the body in general. Norepinephrine tends to be the neurotransmitter that's being used within the synapses. However, a lot of the adrenergic receptors will bind to both of them. So they, the same cell can play two different roles. It can be part of a more directed circuit, part of a synapse, but then if the adrenaline message gets broadcast to the entire body, it will respond to that as well. All right, we'll come back to this a bunch. When we get to the sympathetic nervous system in particular, that's all about adrenergic pathways. Norepinephrine is also involved in things just in the central nervous system that aren't part of sympathetic motor as well. All right. Other biogenic amines. So biogenic amines, again, so catecholamines, couple of enzymatic steps from tyrosine You also have the endolamines. Let's go to the endolamines now. So this is still biogenic amines. So these are derived from tryptophan. Oops. We've already met one of the most famous of the endolamines, serotonin. And we also call it 5-HT for 5-hydroxytryptamine. That's another synonym you often see for it. We'll see it in many different roles as well. It's an important neurotransmitter in your brain, but it's also going to be an important part of hemostasis. Like if you have a tear in your blood vessel, it plays a role in that. It's an important neurotransmitter in your enteric nervous system, just the self-contained neural networks within the walls of your digestive tract. So it's doing lots of different things. So serotonin we will see quite a bit as we go on in the semester as well. So that is the end of the biogenic amines. There are also some neurotransmitters that are amino acids, not the main amino acids that we use to build all of our proteins, but they're amino acids. One that I've mentioned already is GABA, which is gamma aminobutyric acid. But nobody calls it that. Everybody calls it GABA. GABA tends to bind these directly gated channels that open chloride channels. Again, what is that going to do to the postsynaptic membrane if you're opening a chloride channel?
Hyperpolarize?
It's going to take the voltage down. So this is one of the most common inhibitory neurotransmitters in your central nervous system. So when you are activating these GABAergic pathways, you are inhibiting the targets. You are making them less excitable. And this is what we said, like all the benzodiazepines increase the conductance of the GABA channels. That's why the benzodiazepines make you less anxious or fall asleep. Another important amino acid that's one of the main primary excitatory neurotransmitters. So this is inhibitory. One of the main excitatory is glutamate. Glutamate is really common. It tends to open up cation channels that are going to make things more excitable. There are two main versions of glutamate channels that you should probably know because a lot of drugs target these. One of the main ones is NMDA. NMDA receptors. The other one are AMPA receptors. You don't worry about what these, this stands for N-methyl D-aspartate. Kind of the same way we had nicotinic and muscarinic receptors. These just get their names based on some chemicals that will preferentially bind one or the other. NMDA receptors, and ketamine is a NMDA antagonist. It binds to and it blocks NMDA receptors. You know, what is the effect going to be if you are blocking NMDA receptors? Maria?
Inhibitory?
Yeah, so you're going to, since normally these are these excitatory pathways that are transmitting messages from one place to another, you're going to block the transmission of those messages. You know, ketamine is a dissociative anesthetic. The higher the ketamine, people get, you know, the K-hole, people get completely dissociated from actually what their body, being connected to what's happening with their physical body. You know, the people talk about just these experiences of just being floating out in just space. Enough of them, you know, it's used as an anesthetic with kids, or actually it's used as a battlefield anesthetic because it doesn't depress your cardiovascular system in the way most anesthetics do. Most anesthetics kind of take everything really low, and you have to really be careful that the person stays is still breathing and they don't die. But this one tends to be safer because it doesn't last that long, but you know, your cardiovascular functions don't turn off the way in a lot of other anesthetics do. Undergoing anesthesia is scary. If I had to take a class on anesthesia in grad school, and it was really disturbing how it's taking you basically closer to death. and it's actually not that uncommon for things to go wrong. You want to really make sure you have someone who's really good, who's your anesthesiologist if you have to go under. And there's also more and more evidence that that you don't necessarily come back totally intact from really deep anesthesia. People who have multiple experiences of having to go into very deep anesthesia, it seems to actually result in some long-term kind of cognitive effects and things like that. So I would say as public service, if you need to, it might be you need to do it, but if you can avoid deep anesthesia, it's that's something that I've kind of come to a conclusion that you want to kind of avoid it in general, just because it's edgy in general, and it's not even clear that you're going to totally come back to where you were. So glutamate, important. NMDA receptors, important. Let's continue. Peptides. So a lot of important neurotransmitters are just 7 or 8 amino acids connected together, just the correct ones. So these are a lot of the endogenous opioids. The things that are part of those pain pathways, the things that, morphine and all that would bind to, enkephalins and stuff like that. There's a couple of different receptors for this as well. We won't go into the detail of that. A lot of hormones are also just little peptides, like all the releasing hormones released by your hypothalamus that talk to your pituitary gland are just little peptides. Insulin is just a peptide. So a lot of signaling molecules are actually peptides, little chains of amino acids. Others. There's lots of others as well. We talked about adenosine. Adenosine, which is this nucleotide base, also is a neurotransmitter in your brain, right? It depends on the context. Is this something you use to build ATP, or is it something you use as a signaling molecule? Depends on the context. I sometimes use the kind of analogy of, here I am holding my stylus here, and what is this? It can be different things. It can be, depending on the context, I can use it to draw on my little whiteboard here and make cool things. Or I could use it, and it's a good thing to clean the wax out of my ears. It's a cool little tool for that. Or another context, I could use it as a murder weapon and drive it through somebody's heart and take them out. Or what, it depends on how you use it in the context. There's nothing specific about this that makes it a murder weapon or a ear cleaner. It's like the context and how you're using it. It's the same thing with a lot of these molecules. That molecule can play a very different role in very different contexts. So we'll see that a number of times as we continue in the class. The same molecule plays different roles in different contexts. Something like carbon monoxide, which is a deadly poison, we'll talk about, binds your hemoglobin and keeps the oxygen from grabbing. But it can actually be a neurotransmitter. Nitric oxide is a really common neurotransmitter. This is a gas. This is just a little gas. Whenever you need it, you basically turn on enzymes that manufacture it. It comes into existence, and it can just diffuse across the membranes without even needing to have any kind of channels. It's often used as a retrograde neurotransmitter. We've been talking about these messages going, releasing the neurotransmitter, and it goes, binds receptors and continuing the message. Nitric oxide can get produced at the postsynaptic terminal, diffuse backward, and bind receptors in the presynaptic terminal and send messages backwards. So, the reality of what a synapse is and all the possibilities for communication and control there, the deeper you look at any of these things, the more and more crazy and complex everything gets. Nitric oxide is also a really common molecule for vasodilation. We'll talk about vasodilation later on, but nitric oxide is one of the important signaling molecules that results in relaxation of the smooth muscle and allows blood vessels to relax. Anandamide I mentioned before, anandamide. This is actually lipophilic. This is the one that binds the cannabinoid receptors. Again, people were trying to figure out, like, okay, we found a receptor that THC is binding to as people are getting stoned. There's got to be some endogenous ligand. It doesn't make sense that there's some receptor for smoking pot, but it's not used for anything else. So when they looked for some endogenous ligand, they found it. Again, they called it anandamide, Sanskrit for bliss, ananda, because they thought that was funny. And these THC, these cannabinoid receptors, they're all over the frickin' body. They're not just in your brain. They're in all sorts of tissues all over your body, actually. People are still trying to understand fully the role they play in everything. But this is another neurotransmitter. And plus, there's lots of others. But just kind of giving you a sense, there's a lot of different ones. We're going to talk about these a little more. We've talked about this. We're definitely going to be spending a lot of time talking about acetylcholine and the catecholamines when we get into the autonomic nervous system. So just kind of giving you a sense that there's a bunch of neurotransmitters and you should have a sense of them. And there's a lot of pharmaceuticals that are specifically designed to either inhibit or to excite one or another of these receptors to these different neurotransmitters. So that's also when you start getting further down the road and you're going to look at all these different drugs that are available in the pharmacopoeia. There's a lot of pharmaceuticals specifically about being agonists or antagonists to these different kinds of receptors. So it's worth kind of getting familiar with them now. And as you get further down the road in pharmacology and all that, you will meet them in much more depth. Any questions about any of all of this?
What type of neurotransmitters are nitric oxide and those? Are those?
They're gases. They're actually gases.
So they're not. What should we call it? Peptides?
No. It's just a peptide would be a protein, right? So nitric. This is just NO. It's literally nitrogen and oxygen.
Okay.
Carbon monoxide is literally a carbon and an oxygen.
Would adenosine be a peptide?
No, it's a nitrogenous base.
Okay.
It's like, what is it? The purine and a ribose. Yeah, so those, the peptides are amino acids connected with peptide bonds. So a peptide is an a few amino acids connected with peptide bonds. And remember, amino acids are the amine, carboxyl, and some side chain. So then if you connect up a bunch of these with peptide bonds, then you have a peptide. Okay.
Professor, I have one question, and maybe we'll get into this later, but when thinking about how these would work and their affinities of how long they're going, like is it that the neurotransmitters would come in and then continue, like how would it have a response in terms of the timing of it? Does that make sense at all?
Yeah, so there's a bit of a lag. Obviously, you have to get the message hits the end of the synapse or hits the end of the axon, has to cause exocytosis. Then there's a diffusion, there's binding receptors, there's opening channels, and that's on the order of milliseconds for that whole thing to happen. And we're going to talk in a few moments actually about how it is that you have all this complicated control systems when the actual parts that are running the system are so really slow at some core level. There's this kind of massive parallel operation going on where lots of different things are all happening at once. So even though these pathways, like in the context of, again, if you think about computers and digital circuitry, signals are being processed on the order of trillionths of a second, whereas through synapses, it's on the order of thousandths of a second. But you have this very different way of connecting things up instead of doing things more serially, kind of working through the problem really fast, but kind of more in a straight line. You have it's kind of slow, but all these different parts converging in on an answer simultaneously. So you actually have the ability to do kind of complicated control and stay balanced and walk around, even though the system is built with these really kind of pokey subparts.
Thank you.
And again, there's different different axons. There's like the type 1 neurons with like the large diameter, heavy myelination, where, relatively they're fast. You send messages like 100, 150 meters per second. Whereas if you get to like, what is it, type C, like fibers, which are thin, unmyelinated, you're literally sending messages like 1 meter a second, like doop, 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 doop. But you're going to do those in circuits where speed is not of the essence.
And then would it just keep getting released? Like I'm thinking, if somebody's went into like respiratory failure for an asthma attack and you administer epinephrine, and so that there's that response to open the bronchial passageways, right? And then is it just that like it continues?
No, it gets clear. As soon as you have it in your bloodstream, there are a bunch of mechanisms that are going to start clearing it out. We're going to talk about that when we get to the endocrine system, but you basically think about a half-life of any kind of thing that goes into your bloodstream, whether it's some hormone or it's some drug. You always talk about if you take some drug or some hormones released, you think about what is its half-life, which is how long until half of it is usually it's like dismantled by liver enzymes. But so basically, if half-life is T, if we start out with like 100% of however much you added into the person, basically after one half-life, about half of it is gone. But then the processes are working more on the percentage of what's left. So after 2 half-lives, half of that is gone. And after 3 half-lives, half of that is gone. And after 4 half-lives, half of that is gone. So it's this exponential decay. It's not actually cleared out linearly. But with adrenaline, the half-life is minutes. So within a few minutes, most of it is kind of cleared out. So then you're kind of back to normal. But it takes a little while. Like if you get scared and then you realize there's no reason to be scared, it's not like your heart immediately stops beating fast. It's going to keep beating fast until you have brought the level down low enough that it's no longer binding and affecting those receptors that is speeding up your heart. So.
Thank you for explaining that.
Yeah. So yeah, half-life is an important concept, again, in hormones, but also just in any kind of pharmacology. That's the basic way you think about things are constantly getting broken down and removed from your body. It's why you don't get, you can do a couple of tequila shots and get your buzz on, but you don't stay buzzed forever. It's like after a little while, you're not drunk anymore, right? Because you got the same kind of process going on. So we are now going to finally kind of formally lay out our big picture for the nervous system. And we're going to be spending the next few weeks looking at all these different aspects of the nervous system. But it's important at the beginning to look at the big picture. So as we focus in on different parts, you have some context of where are we actually in this whole thing. Again, if you've been in anatomy, this first part should feel familiar. We're going to kind of lay out the overall structure of the nervous system. Again, what are functions of the nervous system? What is it doing?
Sensory
messages.
Say it again.
It delivers messages throughout our body.
So it's delivering messages throughout our body. So it's got a lot of fast communication down the neurons. And to what ends? What kind of messages does it send?
Chemical.
So yeah, it uses like those chemical synapses. What kind of information? What kind of information? Maybe so I should reword.
Like what our body needs to do, or what actions to take,
Right, so
are
part of it is we have kind of what we call the motor division. Motor is about actions. It's about making the body do different things. These are what we often call effectors. You know, the things that you can turn on to do things. What are the different kinds of things you can turn on? What are the things you can activate through the nervous system?
Glands are muscles.
Exactly. So there's glands. You can turn on glands. Maybe I need to salivate because I have some food in my mouth, or I need to secrete some sweat at my skin because I need to cool off. So one of the things you can turn on with the motor division are glands. Other thing is muscles. And of the muscles, you know, there's some of them that you think of that are like, you know, your biceps. So you're moving your body. So some of these are going to be voluntary, the muscles you think about, but a lot of the muscles are going to be, you know, autonomic, you know, the involuntary. I should probably, this is, right? Some of the muscles you are activating are adjusting the diameter of your blood vessels to adjust blood flow and blood pressure. Some of the muscles you're activating are squishing food through your digestive tract. Some of the muscles that you are activating are changing the diameter of your pupils by doing these little muscles in your iris. Some of the muscles are changing the bending on your lens as you focus far and near. So some of the muscles are voluntary, like kicking a ball. Some of the muscles are involuntary, like changing the diameter of your pupil. So this is going to be part of the nervous system, this motor division, the motor division, which are glands and muscles. What other things is the nervous system working with?
Sensory information.
Sensory information, absolutely. So we have this whole sensory division. This is like this motor division. You know, getting information about, and this is both external and internal information. Right? You obviously have like the five senses, like vision and hearing and taste and smell and touch. And we're going to look at all of those in detail. But there's a lot of internal information too, like what is your blood pressure? What kind of food is currently in my stomach? What is the pH of my blood at the moment? Things like that. So there's a lot of information about your internal state that you are not consciously aware of, but that still is being detected and processed by your nervous system. And we'll be talking about that. You know, we saw that already in the reflex lab that we're going to finish up today. This idea of the stretch receptors in your muscles, right? There's little stretch receptors that are telling your nervous system about the stretch of your skeletal muscles. That's part of the sensory division, but not that, it's not something you're consciously aware of all the time. But it's still being used for reflexes to make sure you don't fall over when you're standing up. And then there's a thing, the whole part in between. So you have information about what's going on inside and outside, but then you have to figure out what do we do with it. So that middle part we're going to call integration. Integration is kind of figuring out what is the appropriate response given the current state of the inside and outside world. And then there's usually some kind of response. You have some responsive appropriate. So when we think about the nervous system, these are like kind of the three main parts, and we're going to be looking at all of them. Sensory division is also known as the afferent division. Motor is also known as efferent. You should know these words. The little mnemonic people usually use to remember them is the afferent messages are arriving here. In the integration, this is usually the brain and the spinal cord. The efferent messages, the things that are going to control things, are exiting the brain and the spinal cord. So afferent arriving into the brain and spinal cord, all your sensory information. Efferent exiting with the E. Brain and spinal cord collectively, what do we call them?
Central nervous system.
The CNS, central nervous system. We'll be talking a lot about the CNS, the brain and the spinal cord. And most of your neurons, almost, I think more than 99% of all your neurons are actually just the little inner neurons wiring up little things in all the little circuitry inside your brain and spinal cord. So there's a lot going on in your brain and spinal cord, as you no doubt know. These other two sides, the sensory division and the motor division, collectively we call the PNS, the peripheral nervous system. We'll spend quite a bit of time in here as well, looking at the details of how do you operate skeletal muscles versus autonomic. How do the sensory messages actually get converted from light to an electrical signal? And we got to integrate sensory information, it has to be in the form of electrical signals. So we're going to spend a lot of time looking at how is it that you convert sugar in your mouth to an electrical signal so your brain knows that you're experiencing sweetness? Or how do you convert light landing on your retina to some electrical signal so your brain knows that there's an image in front of your face? So we'll spend quite a bit of time looking at how the sensory division is able to actually take all these different modalities of sensory information and convert them into electrical signals for the brain and the spinal cord. And then we're going to spend time what's going on in the brain and the spinal cord. Then we're going to talk about what are the different major circuitries that control the effector organs, the muscles and glands that are, responses to whatever's going on. There's a little more that we need to do to elaborate on the motor division. So I'm going to kind of take this and we're going to kind of zoom back a little bit, and we're going to kind of, so again, the whole nervous system, if we talk about the nervous system, we talked about the CNS, again, which is the brain and spinal cord, and the PNS. Whack. Which is going to be sensory versus motor. So far, this is what we have been writing on that previous page. Again, remember, sensory is both internal and external. We'll talk about this in more, the interoreceptors and exteroreceptors. Motor. Motor, we need to subdivide. I talked about, voluntary. So one division of the motor is somatic motor. This is going to be the voluntary. This is going to be the part that operates your skeletal muscle. This is when you want to kick a ball or swat a fly or, caress your sweetie's face or whatever. You need to operate some muscles that are voluntary, and there are specific motor pathways, and we're going to see that have specific properties that are different from the pathways that are operating the autonomic or the automatic. The autonomic motor, this is involuntary. This is the stuff that's speeding up or slowing down your heart. This is the stuff that's making you sweat. This is the stuff that is changing the diameter of your blood vessels to change your blood pressure. So this is still running muscles and glands and stuff, but not stuff you're consciously thinking about. And this has two subdivisions, which most people know, the parasympathetic and the sympathetic. Parasympathetic is the classic kind of rest and digest. Sympathetic, people usually think of as fight or flight. In general, they have kind of antagonistic behaviors. If you operate autonomic, parasympathetic, it's the things that tend to be about kind of maintaining homeostasis, slowing down your heart. Sympathetic is that fight or flight, speed up your heart, dilate your bronchoplasty so you get lots of oxygen down into your lungs, dilate your pupils so lots of light's coming in so you can see what's going on. So sympathetic, we're going to look at these in way more detail. But in general, and they're not always antagonistic. We'll talk about that as well. There's more subtlety to it. But it's worth splitting it up like this because we're going to find they're very different in terms of their both neuroanatomy and neurophysiology. And it's really easy to selectively affect one or the other using different kinds of drugs. So it's important to understand how these are functionally different. So you understand, like, what will an anticholinergic what would a cholinergic agonist or antagonist do versus an adrenergic agonist or antagonist based on what we know about the physiology of these different divisions. So we need to think about them separately. They're both motor. Like sometimes people get confused, like, what is the sensory part of the sympathetic? Like that doesn't make sense. Sympathetic is a subdivision of the autonomic subdivision of the motor subdivision. So if we are talking about sympathetic nervous system, no, we're talking about activating muscles and glands. If we're talking about parasympathetic, again, no, we're operating muscles and glands. There might be some sensory information that came in that is resulting in some activity in one of these, but these are specifically motor subdivisions. This weekend, if this is not like crystal clear to you, make sure that you are really clear about this organization here. CNS versus PNS, PNS being sensory and motor, motor being somatic and autonomic, autonomic being parasympathetic and sympathetic. Because we are going to be looking at each of these divisions and drilling down, looking at them in a lot of detail. But if you don't actually have the big picture in mind, you're going to be kind of lost. you're gonna like, okay, I understand this, but I don't actually see how it fits into the big picture. So make sure you know the big picture.